Today we are going to study about the packaging of DNA helix within the nucleus. The nucleus is a very small structure. The nuclei is a very small structure which is present in the cell, and the dimensions are approximately, it is 10 to the power minus 6 meters in size. So it's a very small structure, and the DNA, which is the main genetic constituent, is packaged within that small structure. So if we study about the DNA, then we will come to know how it is packaged inside that. Let us first get the basic facts about it. Now this is the DNA double helix structure. These two strands which run opposite to each other, that is they are complementary to each other. This is the sugar phosphate backbone, which is formed by the sugar molecule and the phosphate. And these two strands run parallel, anti-parallel to each other, ensuring that one end is 5' and the other end is 3', and the complementary strand will be 3' and 5'. Now here, within these two backbones, that is the sugar phosphate backbone, that is the ribbon-like structure which we are seeing over here, we have the nucleotides or nitrogenous base pair, that is A will always pair with thymine and guanine will always pair with cytosine. So they are held over here within this with the help of hydrogen bonds. Now the diameter of a double helix molecule of DNA is 20 Armstrong, and the distance between the two base pairs over here, these are the two base pairs. There are base pairs all over here. I am just showing for this particular region, but it consists of the base pair all over here. Now the distance between the two base pairs is 3.4. This is the distance, and it is 3.4 Armstrong, or in 0.34 nanometers. If we put it in meters, it will be 0.34 into 10 to the power 9 meters. So this is the distance between the two base pairs. Now, this helix or the double helical structure consists of turn after every 10 base pairs. So at every 10 base pair, there is a turn, complete turn, and this is actually the distance between this one turn, complete turn, is 34 Armstrong. Now, considering the number of base pairs of a mammalian cell, this is the distance between the two base pairs. Now considering the number of base pair of a mammalian cell, it is around 6.6 into 10 to the power 9. So this is the total number of base pair which is present in that particular cell. Then if we want to calculate the total length of the DNA, and also considering the distance between the two base pair, it can be calculated by the total number of base pairs, that is 6.6 into 10 to the power 9 base pairs, multiplied by the distance between the two base pairs, which is 0.34 into 10 to the power 9 meters per base pair. And the value comes out to be approximately the total length of the DNA present in the mammalian cell is 2.2 meters. It is an approximate length. Now this length DNA has to fit in the size of a nucleus, which is 10 to the power minus 6. So this requires the DNA to be packaged in a special manner, and this we are going to study in the subsequent videos. Let us first study about the packaging of DNA in a prokaryotic cell. In the prokaryotic cell, the DNA is present but it is not bounded by a membrane. It is having a structure known as nucleoid, where all the genetic constituent is present. So the DNA which is packaged in a prokaryotic cell is packaged as with the help of specialized proteins, and these specialized proteins have DNA over here, and this forms the loop anchor. This is red over here, which is the loop anchor, and this is the loop domain. This structure which is forming the oval shaped structure which is over here is the loop domain. And in the case of prokaryotes, the DNA is supercoiled. The proteins are special proteins, but these are not histones. We will study in the next video the packaging of DNA in the case of eukaryotes, where they are packaged with the help of special proteins called as histones. But the prokaryotic cells do not have histones. They have special other proteins which cause or which hold the nuclear material or the genetic material, that is the DNA, and this DNA is super coiled over here. So in the next video, we will try and understand the structure or the packaging of the DNA in the eukaryote.